Hi everyone, welcome. I hope I'm audible and visible to all of you. Sorry. Yes. So let's start with today's session. Last class we started off. Hi Nitesh, how are you? Hi Diya Rani, hello. Fine. So last class we started up with a new chapter, that is chapter 10 according to your NCRT, and it is basically use of microbes in human welfare. So, as I told you before that, we have always studied a microbe as pathogenic. That means how it causes harm to us. All right. But in this chapter, you will really be amazed to know that how in every single process, in every single industry, these microbes help us. So, how or what all are the steps to get them and in which all industries we use them, I will be discussing it today in detail. So today I will be covering very interesting part of antibiotic production, of how we produce antibiotics with the help of these microbes, along with alcohol fermentation. So how do there are many types of alcohol, okay, and how each of them, like wine के लिए एक microbe होती है, there's a different process for vodka, gin, so all that we will be discussing, and also some other uses of enzymes, or what I should say as biocatalyst. All right, so all this we will be discussing today, and yes, I think these are the major industries we will be covering today. Mainly, we'll be looking into the process of fermentation. And in the last class, we looked at how microbes are used in the household. या everyday use में हम उसे कहाँ कहाँ use करते हैं. Fine. So I hope you find this lecture quite interesting, and please do share the link with all the people you know that they will find this useful. And yes, so I'll start in a minute. So first, let me introduce myself. I'm Janani Saptharishi, and I'm currently a BTech Biotechnology student. I'm student just like you, and I will be teaching class 12 students biology, mainly those who are preparing for their NEET examinations. So, Unacademy has brought you a special feature, and I take the privilege to introduce it. The main features of this are you will get a personal mentor. So, in the iconic subscription by Unacademy, you get the benefit of a personal mentor. That means you will have one-to-one guidance. You'll be guided with the top expert. All right. You'll have a personal mentor who will work on your strength and your weakness, and they will align your concerns and align your learning. Then you'll have live doubt clearing sessions. All right, you can clear your doubts. This is a specialized, customized session made for you. You'll have a parental connect. ठीक है, पेरेंटल कनेक्ट मतलब आपकी जो सारी प्रोग्रेस है इट विल बी डिस्कस्ड विद योर पेरेंट्स. फाइन. Yeah, then you will also avail all the benefits of plus. Okay, so let's just see what all you'll get the benefit in the plus subscription. In plus subscription, you will avail daily live classes. So these classes are very interactive. They are conducted on a day-to-day basis, and they are really beneficial for you all. So all this is done in a very interactive way, and they indulge you in discussions. You can clear your doubts. All right. Then you have live tests and quizzes. So this is very integral for your self evaluation, and you will come to know where you stand in the competition. So with Unacademy, you can prepare for any examination you wish to crack for. All right. So we offer you a structured course for all these, and by structured course, I also mean that we offer you complete syllabus coverage. So complete syllabus coverage, that means see, if you're using my code, all right, to get the subscription, it does not mean that you will avail the contents only of physics, chemistry, and biology. No, that's not the scenario. So when you use my code, sorry, I mean, when you use my code, it is not that you will avail contents only of biology. Instead, when you use my code, you can avail all the contents of physics, chemistry, biology. Like I'm presuming that you are interested in your NEET exam since you're here. So what happens is in these structured courses, you, whatever code you will use, you will get access to all the contents that is related to that particular course, and you'll also get unlimited access. By unlimited access, I mean that one subscription will get you access to all the lectures recorded as well as the live, along with test quizzes, everything. So you can do it at your comfort of device, and we have live sessions every day, and you can watch it at your comfort, at your ease, at your timing. So that is a very big benefit, and also you can make the most after such time, just sitting at home and keep your learning on. You will be on board with the top educators of India, all right, and all these subjects will be covered for you, and we offer you a very, very comprehensive syllabus. So all you have to do is go and download this Unacademy learning application. All right. And once you download it, just install and you get the subscription. You can connect with us directly through the Telegram app also. And we have two features. As I told you, this is a new feature that I introduced. Sorry, this is the new feature that I introduced, the new one, and this is the plus one. So what is the similarity, or what advantage do I get between the iconic? So in both of them, you'll be on board with the top educators of India. In both of them, you will have interactive live sessions. You will indulge in live tests and quizzes, all required for your self evaluation. Then you'll also have a creative corner. But the advantage you will get here is that you will have a personal mentor. This is a more customized one. You will have live doubt, one to one doubt clearing sessions. You'll have a parental connect. So, this is all very integral to discuss your progress along with your parents, and basically to strategize more well to crack the examinations. And you'll also be given a study planner. All right. So this study planner will be completely customized for you, and hence it will help in your learning. एक minute. Huh? In case of any doubts, you can just put a text, drop a text here. I'll keep a track on the comments. Fine. So now coming to the fee plan. So if you wish to go for the plus subscription, you have 3, 6 and 12-month plan. The 12-month plan is the most economical, and at least to crack an examination like NEET, you'll at least need 12 months in hand, no doubt. So the 12-month plan, you are getting 12 months of all the contents for just 15,000 rupees, which makes it very much economical. And now the interesting part is that if you use my code, that is Janani, so you get a 10% off in all these courses. All right. Now, coming to the iconic subscription. Now, if you want to go for the iconic subscription, the 12-month plan is for 28,000 rupees. This is again very beneficial because it is customized for you. And now, if you use my code, that is Janani, the 12-month plan, you will avail it in just for 25,200 rupees. Fine. Now, this is the link for a Telegram channel. Okay, this is all given in the description box below. And what all benefits you get? You get live classes. You can interact with your educator. Alright, you have live poll. You have leaderboards. You can judge your performance. You have analysis. You have test series. Alright, you'll have live doubt clearing sessions. And I've already made you aware of the steps on how you go about it. Just install, and you have to select your class, what exam you're interested to crack, and your goal. So here your NEET is your goal, and your class is your class 12. So this is the subscription plan which we have already discussed. 12 month is 15,000 rupees, and if you use my code, you will get an 10% off. So now, if you want to attend my special classes, all right, which is absolutely free, how do you do that? Just download the app, set your goal similar, just and you will select the criteria of special, all right, and you use my code, that is SP Janani, and you'll get an invite code to the and you'll get the access to my free plan. So this is how you join a special class, that is a theory lecture. And if you want to join the quiz, again all the things is same, but you only have to select your the join quiz, and you need to enter my code, that is SP Janani. All right, with the help of this code, you can gain access to all my contents. So these two are already done. So the upcoming class is tomorrow between 3 to 4 p.m. And you will be having a quiz session mainly on immunity, that is from your human health and diseases. इसी का एक part है, उस पे कल quiz होगा. So those who are interested to attend this free quiz session, you can use this code, and we see you tomorrow at between 3 to 4. Fine. So now let us move to the main point of today's lecture, that is how we can use microbes in our industry, how integral part, or what how so important it is, and how all the process is carried out, how the fermentation is carried out. All right, so we will be discussing that. Microbes are basically to produce industrial products. तो क्या होता है कि सबसे इम्पॉर्टेंट जो स्टेप होती है इंडस्ट्री में, वो होता है हमारा फर्मेंटेशन. फाइन. सो even in the industry, microbes are used to synthesize a number of products that are valuable to human beings. That is, we know, and this is how large processes in industries are carried out. All these fermenters or fermentation tanks, or which are commonly called as bioreactors, they are very widely used. Why we use it? To manufacture or make a product that is valuable to us humans. All right. And the example of these products, so हम क्या क्या products बनाते हैं? We make antibiotics, mainly healthcare sector, pharmaceuticals में काफ़ी use होती है. Then any beverage, that means alcoholic beverage भी ले लो आप, your wine, whiskey, brandy, gin, everything is processed with a process called as fermentation. This is very very important. Then even your fruit juices, all right, your cold drinks, everything has to undergo the process of fermentation. So we'll be learning about these two mainly today, your alcohol beverages, and your antibiotics. Fine. So last class I discussed in detail about the process of fermentation. So just to brief you again about it. So fermentation is basically a catabolic process. ठीक है. जनरली हम क्या करते हैं यहाँ पे, हम एक प्राइमरी मेटाबोलाइट को, ठीक है, we convert it into secondary metabolite. ये कैसे हो पाता है? हम प्राइमरी मेटाबोलाइट को सेकेंडरी में कैसे कन्वर्ट कर सकते हैं? ऑल राइट. सो प्राइमरी मेटाबोलाइट का एग्जाम्पल मैं क्या लूँ? जैसे कोई भी मेरा शुगर हुआ, कार्बोहाइड्रेट हुआ, फाइन, this all is the example of my primary metabolite. So generally that is how you see fermentation, right? Breakdown of sugars into alcohol. तो ये बहुत बेसिक डेफिनेशन है, बट इट इज नॉट ऑलवेज अल्कोहल. I discussed in my previous class also, you can go and see it in my playlist, that ये जो है it can be even like any gas or any of the acid like citric acid, glutamic acid, ये सब fermentation से ही produce होता है. Alright, so it can be any of the acid, alcohol. By acids I mean such, okay, like I said the example, citric acid, glutamic acid, ये सब acids हम produce कर सकते हैं. It can be alcohol, or it can also be any of the following gases. All this can be your byproducts. Now, how this is carried out? This is basically carried out from the action of your microbes. ठीक है. बेसिकली माइक्रोबियल एक्शन से आपका ये कन्वर्ट होता है. एंड यू ऑल्सो नीड समथिंग कॉल्ड एज अ nutrient medium. Now, this nutrient medium can be anything like carbohydrate or water, or or you can just simply, it can be any medium of your interest or what according to your requirement, I would say. Fine. So, yes, so all that you need to do, it is a catabolic process. Fine. Converting some a primary metabolite to a secondary with the growth, with the action of microbes, very important, and you will have a byproduct. So, generation of useful products on an industrial scale requires growing microbes in very large vessels called as fermenters. ठीक है, तो ये फर्मेंटर्स क्या होता है, जैसे यहाँ पे दिखा ना, एग्जैक्टली वैसे दिखता है, वेरी लार्ज वेसल्स where you have all the process going inside. This has many inlets, outlets. I'll draw a diagram for you. ये जो diagram है ना इसका, आपके बायोटेक वाले चैप्टर के एकदम एंड पार्ट में है. That diagram is very important, and that is how your fermenter look. It can also be called a fermentation tank, or more widely known as a bioreactor. Fine. Very widely used in all the industries. All right. And I don't think I have. Yes, so let me first draw the diagram for you all. So I'll do my best that how detailed can I go here, but basically refer it with the chapter of biotechnology. उसमें बहुत अच्छी डायग्राम दी हुई है. ठीक है. सो एक ऐसा एक टैंक होता है बेसिकली. ठीक है. एंड सबसे इम्पोर्टेंट होती है यहाँ पे, सबसे पहले तो मोटर होगा. एंड ये मोटर क्या करेगा, ये अटैच होता है एक शाफ्ट से. ठीक है. वो शाफ्ट हमारे किस काम का है, हाँ, ऐसे एक shaft होगा. All right. And here there are many blades attached, somewhat like this. Fine. So this basically turns round. All right. And this is known as your steerer. Fine. Steerer किस काम में आता है, basically your steerer is for your agitation. Fine. It basically keeps on turning the material or the content around it. So this might have some, yes. All right. Then after that, this is the main part. ठीक है. यहाँ पे अब जो, because ये steerer continuously turn कर रहा है, mix कर रहा है, आपके microbial action को इसमें अच्छे से हेल्प करेगा. ठीक है, होने में. एंड इट विल cause, eventually it will lead to fermentation. See, fermentation is nothing but the growth of microbes. But you see, if there is no stirrer, then that microbe will not get completely, mixed with the entire content. So that makes it very integral. There will be a constant mixing, agitation process. This also increases the this also increases that the microbial action, and it will lead to or earlier fermentation, or the appropriate one as we need. So there will be, first of all, the inoculation pipe. ठीक है. इनोकुलेशन पाइप क्या होती है, जहाँ से हम अपना मटेरियल ऑफ इंटरेस्ट यहाँ पे इनोकुलेट करते हैं, एंड हमारा बैक्टीरिया भी इसके अंदर हम इसको इनपुट करते हैं. ठीक है. एंड हियर यू विल हैव, या, सो बेसिकली सबसे पहले तो यहाँ पे स्टेराइल एयर का आउटलेट रहेगा. एंड यू विल ऑल्सो हैव योर हार्वेस्टिंग पाइप मतलब जो रेडीमेड मटेरियल जो भी बन चुका है उसको हम जहाँ से इस टैंक से बाहर निकालेंगे. फाइन. सो दिस इज क्लियर. आई टेक अ डार्कर कलर ओनली. या. फाइन. अदर देन दिस व्हाट यू विल हैव समथिंग कॉल्ड एज अ फोम ब्रेकर. ठीक है. फोम ब्रेकर क्या करता है कि अभी क्योंकि ये कंटिन्यूस एजिटेशन मोड में है, कंटिन्यूसली टर्न हो रहा है, सो देयर विल बी अ लॉट ऑफ फोम दैट इज फॉर्म्ड हियर. So this foam breaker is basically to check on that over foam या वो इकट्ठा froth इकट्ठा नहीं होना चाहिए, इसीलिए we use it. Then you will have a pH probe that will maintain your pH. You will have a temperature probe that will maintain your temperature. Then you will have an oxygen sensor, all right, which will basically sense your oxygen levels. And you will also have something known as a compressed air inlet. ठीक है. Compressed air inlet. So this basically makes sure that you're getting the right amount of oxygen and it is going inside this. And you'll have a sampling pipe here. Fine. So this sampling pipe is basically from where you can collect your samples. So I hope this structure is clear. So this is just an overview. I'm just making you familiar with all the parts that are involved in a bioreactor, or basically how is an outline of an fermentation tank. Okay, in case you have any doubts, you can just pin it up. कभी कभी मुझे लेट दिखाई देते हैं कमेंट्स. Anyways, so ठीक है. So we'll start with fermented beverages. All right. So fermented beverages में क्या आता है? Microbes, especially yeast, have been used for the production of beverages. As I told you, mainly alcoholic beverages fermentation. Alcohol is a very big industry, very much in trend, or I should say that a large industry which makes a lot of profit and which is in demand. ठीक है, तो ये काफी in demand होती है. So we need such huge tanks, or I should say that such huge fermenters that we can match the need with the supply. All right. So alcohols में किन किन के बारे में अगर मैं precisely बात करूँ तो we require the industrial value सबसे ज्यादा होती है. Hi Angel Sheikh, how are you? Welcome. So, alcohol की अगर मैं बात करूँ तो wine, whiskey, brandy, rum. For all these, we require a large amount of fermentation process. All right, and we will see कि हर इसके लिए requirement कैसी अलग अलग है. All right, so this is wine. This is, I guess, this is whiskey. ठीक है, this is gin, I think. Yeah. So the same yeast that is Saccharomyces cerevisiae. ठीक है, Saccharomyces cerevisiae हमने कहा था ब्रेड के फर्मेंटेशन भी यस ब्रेड मेकिंग में भी यूज होता है. ये हमने हाउस होल्ड वाले इसमें पढ़ा था. नाउ इंडस्ट्रियल लेवल Saccharomyces cerevisiae को हम कहते हैं ब्रूअर्स यीस्ट. प्लीज रिमेंबर दिस टर्म. Alright, this is a line taken as it is from your NCRT, very important, and it is used for fermenting malted cereals, fruit juices to produce ethanol, and this is how it is used for your alcoholic production. ये क्या करता है आपके malted cereals यानी कि कोई भी cereal हो, even your molasses जो sugar cane से हो, उन सबको and your fruit juices also. बेसिकली वाइन में जो होता है ग्रेप जूस को ही हम फर्मेंट करते हैं. ठीक है, तो इन सब में ये यूज करते हैं. अगर मैं ये ब्रूअर्स यीस्ट में बनाऊं, so it will ferment and give me the byproduct as ethanol, that is your alcohol. Fine. Hi Akshay Bhomik, how are you? Welcome. ठीक है. So दिस इज ब्रूअर्स यीस्ट ऐसे भी अवेलेबल होता है. मैंने इसलिए पिक्चर डाला है ताकि आपको भी पता चले कि मार्केट में कौन सी चीज कैसी दिखती है, हाउ इजी इट इज अवेलेबल और हाउ difficult it is to get it. All right, so it can use in all these products. So even see, as I said that cereals को भी हम ferment करते हैं. जैसे जो राई होती है ना, it is very बहुत महंगी क्रॉप होती है, उसके fermentation से जिन बनता है. All right, so that is how the process is carried out. And yes, juices पे तो आपको पता ही होगा, wine is made up of your grape juice. So this is how the fermentation carries out. ये तो बस easy way में बोल रही हूँ, but when you see it in the industrial way, the bioreactor, you know, I just drew the diagram of the bioreactor, and such huge tanks are used, and it is a complex process but very widely used. And इसके fermentation से क्या बनता है? इन सबके fermentation से बनती है इथेनॉल. सो इसको आप एक मैकेनिज्म की तरह देख लीजिए, या ये आप याद रखना कि इन सबसे इन अल्कोहल इंडस्ट्री में कैसे होते हैं माल्टेड सीरियल्स, जूस, जो भी आपका प्राइमरी मेटाबोलाइट हो, उनको फर्मेंट करके क्या बनते हैं? इथेनॉल. प्लीज रिमेंबर. एंड व्हाट इज यूज्ड? ब्रूअर्स यीस्ट, दैट इज योर Saccharomyces cerevisiae is used. See, depending on different alcohol, the genus may change, but the species Saccharomyces remains the same for all. तो Saccharomyces species ही mainly सबसे important होती है आपकी alcohol industry में. It is asked many times in your MCQs, so please remember. All right. So now, depending upon the type of raw material used for fermentation and the type of processing which is involved. So type of processing means दो type होंगे, एक with distillation and one will be without distillation. So different types of alcohol drinks are obtained. ठीक है. वाइन एंड बियर तब प्रोड्यूस होते हैं जब वो विदाउट डिस्टिलेशन वाली प्रोसेस इनमें स्किप हो जाती है. फाइन. एंड Whiskey, brandy, rum are produced by the distillation of the fermented broth. Fine. So you have to remember what alcoholic beverages are distilled and what alcoholic beverages are not distilled. Fine. Now, uh, not only this, I want to add many points here. So basically, एज आई सेड दैट ये तो आपका है ही बेसिक्स में कि कौन से डिस्टिल्ड है कौन से नहीं है ऑलराइट सो या नाउ इफ यू वांट टू सी Saccharomyces cerevisiae जो हमारे पास अवेलेबल होती है आई एम नॉट राइटिंग द एंटायर नेम प्लीज बेयर विथ मी दिस इज योर ब्रूअर्स यीस्ट ठीक है जो आपके बियर में एंड ऑल यूज होती है फाइन this is used in your production of your beer. Then you have something known as Saccharomyces ellipsoids. All right. So it is ellipsoides. ये जो स्पीशीज है ये एक्सक्लूसिवली यूज होती है आपके वाइन प्रोडक्शन में ऑलराइट प्लीज रिमेंबर दिस देन योर फर्मेंटेड राई ऑलराइट राई इज अ टाइप ऑफ अ cereal so this is used for the production of gin then if you have your molasses को ferment करके बनती है rum ठीक है so this can be asked in many of your advanced examinations like your aims and all so basic फर्मेंटेशन किससे कौन से ऑर्गेनिज्म से क्या बनता है बेसिकली कौन से स्पीशीज से क्या बनता है आपको याद होना चाहिए बट मेनली योर Saccharomyces species that is your brewer's yeast is only used fine so now let us move forward towards the antibiotic production so antibiotics is very largely used, and you cannot imagine uh, a life, not a life like you cannot imagine anyone's survival because without antibiotics. Antibiotics are itne we are dependent on them that even for a mild fever, a dose of antibiotic is definitely recommended. Fine. So it will be very difficult antibiotic के बिना survive करना, especially in today's world. And yes, and antibiotics is also again a boon that we have got from microbes. Okay? And it is regarded as one of the very significant discoveries of the 20th century. All right, and काफी significant माना जाता है medical history में. And this is basically anti is a Greek word that is against. And yes, so basically, what do I mean by antibiotic? एंटीबायोटिक्स यानी कि एक माइक्रो ऑर्गेनिजम्स या एक लिविंग माइक्रो ऑर्गेनिजम्स ओके I'll write it as microbe when it inhibits all right or I should say stops the metabolic activity of another organism ठीक है ये होती है डेफिनेशन मेरी एंटीबायोटिक की यानी कि एंटीबायोटिक बायो मीन्स लाइफ ठीक है सो इट इज बेसिकली इनहिबिटिंग द ग्रोथ और स्टॉपिंग द मेटाबॉलिक एक्टिविटी ऑफ अनदर ऑर्गेनिज्म ठीक है सो बट दिस एंटीबायोटिक प्लीज कीप इन माइंड ये दूसरे सिर्फ हमारे टारगेट ऑर्गेनिज्म पे ये ऐसे उनको इनहिबिट करना चाहिए इट शुड कॉज no harm to the host all right so if I am taking an antibiotic it should only affect the pathogen and not me so that is some criteria that we need to look upon with the antibiotics so anyways so yeah. So antibiotics are chemical substances which are produced by some microbes and they can kill or retard. That means inhibit the growth of another disease causing microbes. तो एक microbe दूसरे microbe को affect कर रहा है. So this is how yes we are commonly familiar with the commonly used antibiotic that is penicillin. So penicillin की discovery भी ऐसे ही हुई थी ठीक है. So Alexander Fleming is the one who discovered penicillin. So if the question comes as who discovered the first antibiotic, it is Alexander Fleming. But please keep in mind that if a question comes that who gave the term of antibiotic, it is not Alexander Fleming. Please don't select this option. This is basically the term given by someone called as Waxman. All right. So this person is the one who coined the term antibiotic. He did not discover it first. He just gave the term and he discovered the species of streptomycin. ठीक है, ये भी एक बहुत widely used antibiotic है. Alright, so he basically found out the uh, species of streptomycin. Fine. So penicillin was the first discovered one, and what happened? It was found by accident. I'm sure everyone must be knowing the story. So what happened that while working with the Staphylococcus bacteria, once he observed a mold growing on this culture plates. Basically, fungus growna start हो गया था Staphylococcus bacteria के plate में. And around this fungus, the Staphylococcus bacteria could not survive. बेसिकली क्या हो रहा था कि वो जो फंगस जो ग्रो हो रहा है दैट वाज रेस्ट्रिक्टिंग और इनहिबिटिंग द ग्रोथ ऑफ माय बैक्टीरिया. सो व्हाट ही फाउंड आउट दैट ये जो फंगस है वो मेरे बैक्टीरिया को ग्रो होने नहीं दे रहा. सो ही फाउंड दैट इट वाज ड्यू टू अ केमिकल प्रोड्यूस्ड बाय द mold. So that is what happens, right? So any antibiotic we take, that particular microbe, it we are interested in the chemical that it is producing. So that chemical is basically has the power that it will restrict the growth of the pathogen. ठीक है, तो hence he named it as penicillin after the mold Penicillin notatum. Fine. So that antibiotic was called penicillin because of the mold that was growing, and उस mold से chemical निकला जो bacteria की growth inhibit कर रहा था. Alright. So this is Alexander Fleming. The full potential of penicillin for established by the efforts of Ernest Chain and Howard Florey. So another two scientists are involved. These two, Ernest Jane and Howard Florey. So yes, this is again given in your NCRT that these antibiotics are widely used during your World War II and even during the outbreak of plague. Fine. Okay. So, yes, Fleming, Chain, and Florey, all these three were awarded the Nobel Prize. All right, and yes, after penicillin, there were several other antibiotics that were also purified from these microbes. So, one of them, as I said, was streptomycin. Streptomycin is again very widely used, and this streptomycin species, we literally see there are almost 700 antibiotics that we have today, and streptomyces species alone gives us more than 40 antibiotics. Fine? Yeah. Then you have gentamicin, then you have neomycin. Okay? Now, antibiotics have greatly improved to treat deadly diseases such as plague, whooping cough is your kali khasi, diphtheria, and your leprosy. In सब के लिए antibiotics को बहुत widely use किया गया है. And in today, we cannot imagine the world without antibiotics. So, before moving to the next one, I need some extra points that I need to cover. Especially, I wanted to speak about the term of antibiotic resistance. We have managed to develop antibiotics, but have you ever imagined that अगर एक antibiotic ही मेरे bacterial growth को कम कर रहा है या effective है इतना, तो why do I have 700 antibiotics? Fine, this is because some antibiotics have gained resistance. So how is that happening? Before that, let me tell you that there are two types of antibiotics. So one is your broad spectrum antibiotics. Okay? Another is your narrow or specific antibiotics. Specific antibiotics. ठीक है, this is your broad spectrum. ठीक है, broad spectrum antibiotics में क्या होता है that it is affected in a wide range of pathogens. ठीक है, ये काफी एक बड़ी स्पेक्ट्रम होती है इनकी. That means it can be used for a number of pathogens. But in your narrow or your specific antibiotics, what happens that they are only used for the action or for the treatment of one pathogen or very few. Fine. So this is the example. This is the definition. Basically, हाय परमेश्वर मुंडा, वेलकम, गुड इवनिंग। तो ये आपकी दो टाइप की एंटीबायोटिक की क्लास बन जाती है, ब्रॉड एंड नैरो स्पेक्ट्रम। ठीक है। अ, और भी जैसे मैंने चीजें बताई थी एंटीबायोटिक रेजिस्टेंस के बारे में। सो before that, what do you think that how is the mode of action? Okay, mode of action of these antibiotics. So, by mode of action, I mean that how these antibiotics are effective towards controlling or killing the pathogens or the microbes that are harmful to us. So they follow a wide range of mode of action. Among that, the penicillin, the streptomycin, or such most frequently used mode of their action is the disruption of cell wall. Fine. Disruption of cell wall. ये कौन करता है? ये है पेनिसिलिन का मोड ऑफ एक्शन। ठीक है, नॉट ओनली पेनिसिलिन बट ऑल्सो सिफेलोस्पोरिन। फाइन। सो व्हाट इज डिसरप्शन ऑफ सेल वॉल? ये बेसिकली आपके जो पैथोजेंस है उनके सेल वॉल को सिंथेसाइज भी नहीं होने देता, एंड इट विल स्टार्ट ब्रेकिंग दी सेल वॉल। अब सेल वॉल डिस्टरप्ट हो गया तो उसकी न्यूक्लियस या अंदर के कंटेंट्स एक्सपोज हो चुके हैं, मतलब उसका प्रोटेक्टिंग कवरिंग निकल चुका है। So now it is very easy for the body or the antibiotic to go and directly kill the affecting microbe or the pathogen. So that is what is the mode of action. It can be even due to it stops the process of translation. ठीक है, या फिर ये आपके बॉडी के 30s 50s राइबोसोम्स को अफेक्ट करता हो, या इवन stopping the formation or disruption basically of your plasmonemma. तो ये सब मोड ऑफ एक्शन होती है आपकी एंटीबायोटिक्स की। फाइन। हाय पुष्पेंद्र सिंह, बहुत दिनों बाद, गुड इवनिंग, कैसे हो? ठीक। सो एक एंटीबायोटिक कहलाने के लिए क्या क्या प्रॉपर्टीज होती है, या आइडियल एंटीबायोटिक्स की अगर मैं बात करूं, सो हाउ कैन आई से दैट इट शुड क्वालिफाई एज द टर्म, और इट शुड क्वालिफाई एज एन आइडियल एंटीबायोटिक्स? Alright, so first of all is that it should be harmless to your host. Harmless to the host. सबसे पहली बात, if I have a disease, all right, and I am taking an antibiotic, मुझे उससे कुछ साइड इफेक्ट या हार्म नहीं होना चाहिए। ओनली द पैथोजन शुड बी किल्ड। ऑल राइट, ओके, दैट्स नाइस। ठीक है, तो सबसे पहली बात, इट शुड बी हार्मलेस टू द होस्ट, और आई कैन ऑल्सो से दैट इट शुड बी मिनिमम साइड इफेक्ट्स। ठीक है, सबसे पहले एंटीबायोटिक में तो ये होना चाहिए, एंड इट शुड स्टार्ट गिविंग इट्स इफेक्ट फास्ट। दैट मीन्स उसकी जो एक्शन होनी चाहिए दैट शुड बी फास्ट, देन आई हैव फास्ट एक्शन। एंड another very important point that it should be harmless to my gut bacteria. All right, so in my gut, in that is my digestive tract, I have a whole microbe, or E. coli is surviving in my gut bacteria. There are many types of microbes. So ये जो है मेरा एंटीबायोटिक जो मैं कंज्यूम कर रही हूँ, that should never harm my gut bacteria. ठीक है, वरना मेरा अंदर का एनवायरनमेंट जो है वो डिस्टरप्ट हो जाएगा, वो हिल जाएगा। So it should not hamper my gut microbiome. So that is another main point. So all these yes, and yes, it should also cover a wide range of pathogen. ये हो तो अच्छा है, wide range of your pathogen range should be wide. ठीक है, इसीलिए ब्रॉड स्पेक्ट्रम एंटीबायोटिक ज्यादा प्रेफर होते हैं, बट स्पेसिफिक भी काफी काम आता है। सो बेसिकली दीज 4 points आर द मेन रिक्वायर्ड वन, एंड यू शुड रिमेंबर देम, that this is required for something to call them as an ideal antibiotic. Then when I come to resistance, I'd asked one question that अगर हमारे पास इतने efficient हम discover कर चुके हैं antibiotics, so why do we have 700 antibiotics that we have today? So this is basically, see, your the antibiotics, they have a particular mode of action that and all we discussed. But what is happening that your microbe, in some or the other way, they have a mechanism and they modify themselves, and then they get resistant. ठीक है, एंटीबायोटिक रेजिस्टेंस ये बहुत इंपॉर्टेंट टर्म है, बहुत आजकल इसके बारे में बात होती है. दे आर गेटिंग रेजिस्टेंट टू माई एंटीबायोटिक्स, एंड ये रेजिस्टेंस की वजह से आई एम नॉट एबल टू टैकल और ओवरकम द इन्फेक्शन. So what is basically happening? So there it can be basically like resistance development हो जाए उनके gene में, that is present in their plasmids. Okay, or or even what happened that they are changing their I'll just write it with the another color. ये resistance वो हमारे pathogen कैसे कर रहे हैं? It can be because they have changed their cell wall composition. ठीक है, सेल वॉल कंपोजिशन अगर चेंज हो जाए, तो अभी जैसे कि मैंने कहा था कि दे कैन नॉट डिसरप्ट देयर cell wall, or there can be a formation of a very protective covering called as mucilage. Mucilage एक और protecting covering आ गया, so it is now very difficult to go and break that pathogenic cell, if I use very simple term. Or the best they can do, they can modify the एंटीबायोटिक केमिकल इटसेल्फ. ठीक है, वो क्या करता है, केमिकल को ही मॉडिफाई कर देता है. अब जब केमिकल ही मॉडिफाइड हो गया, सो नाउ इट इज वेरी मच डिफिकल्ट for my antibiotic to go and make its effect on them. So this all is hi Vikas Pandey, आप यहाँ कैसे, वेलकम, थैंक यू फॉर ज्वाइनिंग. ठीक है, तो ये सब जो है एंटीबायोटिक्स अपने आप को इतनी, सॉरी नॉट एंटीबायोटिक्स, योर माइक्रोब्स मेक देमसेल्फ सो मच रेजिस्टेंट टू द एंटीबायोटिक्स कि हमें एक न्यू स्पीशीज या एक न्यू एंटीबायोटिक लाना पड़ता है. सो हेंस व्हाट इज हैपनिंग, वी आर जस्ट इंक्रीजिंग द डोसेज and also the consumption of antibiotics, and there is more and more microbes that are getting resistance to it, and this is posing a great threat. So this is a very wide topic, we can go on speaking, but this is all you need to remember. And yes, I think now we can move forward with bioactive enzymes or molecules. So basically, what do I mean by bioactive molecules, or how chemical enzymes and these are used in my industry? अब हम उसके बारे में पढ़ेंगे. हम दो चीजें कवर कर चुके हैं, very important, that is alcoholic fermentation and your antibiotics. This for my two main topic of interest, very important from this chapter. Now the last part is of chemical enzymes and other bioactive molecules. So microbes are used in my commercial and industrial sectors, and now it is also used for certain chemicals like organic acids and enzymes. Alcohols तो हम देख ही चुके हैं. So the example of acid production, मैंने जब भी fermentation की process भी describe की थी, तो I told that it is a basically conversion from primary to secondary metabolite. Secondary metabolite, I had specifically mentioned that it also can be an acid. By acid, I meant the following. So your asparagus niger is a type of fungus that will help in my citric acid प्रोडक्शन. सिट्रिक एसिड कहाँ कहाँ यूज होते हैं, ये यूज होती है मेडिसिनल में. आपको जो भी विटामिन सी के इतने सारे टैबलेट्स मिलते हैं, इम्यूनिटी बूस्टर्स मिलते हैं, उन सब में सिट्रिक एसिड यूज होते हैं. इट इज यूज्ड वाइडली इन द डाइंग इंडस्ट्री. ठीक है, डाइंग इंडस्ट्री यानी कि टेक्सटाइल इंडस्ट्री में कलर के लिए काफी ज्यादा यूज होती है. एंड इट इज ऑल्सो यूज इन माई फ्लेवर इंडस्ट्री. ठीक है, सिट्रिक एसिड बेसिकली इट हैज अ वेरी सोर टेस्ट, एंड ये बहुत वाइडली हम यूज करते हैं इनके फ्लेवर्स इन माई इन द कैंडीज एंड chocolates or such products. All right, so then you have the acetic acid production. Acetic acid is again very widely used in your pharma. ठीक है, ये प्रोड्यूस होते हैं एसिटोबैक्टर एसिटी से. ये आपको जरूर, मैं जितने भी स्क्रीन पे आपको दिखा रही हूँ, ये माइक्रो ऑर्गेनिज्म एंड किस टाइप ऑफ माइक्रोब से क्या प्रोडक्ट बनते हैं, ये टेबल आपको जरूर याद होना चाहिए. देन एसिटिक एसिड इज इवन यूज्ड वाइडली इन योर कलरिंग एजेंट्स. ठीक है, then you have your Clostridium butylicum, that is the manufacturing of your butyric acid. And lactobacillus तो आप जानते ही होंगे, lactic acid बनाता है, lactic acid milk industry इन सब में बहुत highly use होती है. Then even your probiotics, lactic acid is highly used. ठीक है. uh, yes, I think that is it for this one. That means your acid production. Now, yeast, that is your Saccharomyces services used for the commercial products. That means जो भी अल्कोहल आप कंज्यूम करते हो ना, उन सब के प्रोडक्शन, that means इथेनॉल में वो हेल्प करता है. We have already discussed that how इससे बियर, वाइन, रम एंड आई गेस जिन, इन चार के बारे में हमने डिटेल में बात की. ठीक है. Then, microbes is also used for production of enzymes. Enzymes is your biocatalyst. Enzymes are very very important. ठीक है. टाइड आपने जरूर देखा होगा, टाइड रिन जो भी मिलता है, तो इन सब में यूज होती है लाइपेजेस. ठीक है. लाइपेजेस क्यों डालते हैं? लाइपेजेस जो होती है ना, लाइपेजेस मीन्स लिपिड डीजेनरेटिंग और लिपिड डिग्रेडिंग. ठीक है. एंड हमारे जो भी ऑयल, जैसे कि हमने कोई भी खाना खाया, उसमें क्या होता है कि हमारे ऑयल के जो भी स्टेन्स होते हैं उनको जाके डिग्रेड करते हैं लाइपेजेस. ऑलराइट, सो हेंस इट इज वेरी इंपॉर्टेंट इन रिमूविंग द ऑयली स्टेन्स इन योर लॉन्ड्री. फाइन. देन वी हैव पेक्टिनेसेस एंड प्रोटीएसेस, यानी कि प्रोटीन डिग्रेडिंग एंड पेक्टिन डिग्रेडिंग एंजाइम्स. ये बेसिकली हमें यूज होता है व्हेन वी नीड टू क्लैरिफाई योर फ्रूट जूसेस. ठीक है. कोई एक्स्ट्रा प्रोटीन या कुछ पेक्टिन रह गया हो तो उन सब में ये काफी हेल्प करता है इन योर बॉटल्ड फ्रूट जूसेस. देन यू हैव योर स्ट्रेप्टोकाइनेज, वेरी वेरी इंपॉर्टेंट, इसको आप जरूर याद रखना. लाइपेसेस एंड प्रोटीएस तो याद रह ही जाते हैं. स्ट्रेप्टोकाइनेज इज a bit of more importance. Okay, so it is basically produced by the bacteria streptococcus and it is modified by genetic engineering. So what it is used at? So it is used as a clot buster. Basically clot buster, if any of the arteries are blocked, it is very important or required to remove that clot. So yes, removing clots from the blood vessels of the patients who have myocardial infarction, that means a heart attack. ठीक है. तो ये किस वे में प्रोड्यूस होता है? यस, अनदर बायोएक्टिव मॉलिक्यूल इज योर साइक्लोस्पोरिन ए, that is used as an immunosuppressive agents. That means you use it in your organ transplant. ठीक है. Whenever you are transplanting an organ from the donor to the recipient, you need to suppress the immune reaction so that they will not affect or they will not start attacking the transplanted organ. So that immunosuppressive agent that is used is your cyclosporine A. Fine. And this you get it from the fungus, or this is produced from the fungus of Trichoderma polysporum. So you can see that this chapter basically has some information about all the biotech part, then something you have from the plant section, then you have this immunology section. So this is basically a mixture of many chapters and how in a large scale you see it. ठीक है. Then you will have the production of glutamic acid, gluconic acid. ये भी asparagus niger ही produce करता है and it is the calcium source, gluconic acid. ठीक है. ये calcium का source बन जाता है for your lactating mothers and also the newborn babies. So I think other than that we have, yes, so one last, the stains produced by the yeast Monascus purpurus have been commercialized as blood statins. Okay, not stains. Sorry, that's what I was thinking. Why stains? Fine. The statins produced by this yeast have been observed in blood cholesterol lowering agents. This is not very important, but the most important is your streptokinase and your cyclosporin A. Please remember these two. Okay. And yes, streptokinase is your clot buster. That also we have discussed. So I think that is it with the industrial production. We have discussed about 3 industries. Actually more than 3, but अगर मैं उसे summarize करूँ, सबसे पहले तो आता है आपका alcoholic industry in detail. Then we discussed about the antibiotic production, your pharma, and then we discussed about how enzymes and bioactive molecules are used. So please be clear with it, and tomorrow we will be discussing about the wastewater management. So yes, so this is one thing that I missed out, that is statins act as a competitive inhibitor for enzyme responsible for synthesis of cholesterol. ठीक है. तो ये हुआ. And one last thing, yes, so you can join me tomorrow. Wait, huh? So I wanted to tell you all that you can join with me in any of the special classes. All right, and yeah. So all you need to go is download the app, select your goal, and you have to use my code, that is SP Janani. ठीक है, तो कल भी एक क्लास होने वाली है, that is from 3 to 4. ठीक है, and it will be a quiz basically on this section of immunity. इस पे quiz होने वाला है. My code is SP Janani. तो ये बिल्कुल फ्री है, आप इस क्विज में मेरे साथ ज्वाइन करना. ठीक है. So yes, you can use a referral code for the plus में referral code Janani है, you'll get a 10% discount there. So thank you so much for watching. Please like, share, subscribe. My code is, again I'll write it down, it is Janani. Please share the link with whomever you feel this will benefit and be helpful to. And you can also join the Telegram group. Thank you so much for joining in. Thank you. Bye.